cultivar or clonal development plays a crucial role in propagating ornamental and fruit trees, ensuring that desirable traits are preserved in new plants. This practice underpins sustainable agricultural methods. The total potency in plants allows the regeneration of an entire organism from any single cell or tissue, underscoring the remarkable attributes of plant biology, which starkly contrasts with animal reproduction. Adventitious roots and shoots are vital in vegetative propagation, as they can form from cuttings, facilitating effective plant reproduction. Familiarity with these processes aids in enhancing gardening practices. Tissue culture exemplifies a method to generate whole plants from a mere piece of carrot, illustrating plants' extraordinary regenerative prowess. This process further emphasizes the distinctiveness of cloning in the plant kingdom. Through vegetative propagation, maintaining plant clones ensures the preservation of specific traits across generations. This technique is imperative for the cultivation of fruit trees and other highly valued crops. Moreover, this method allows for the successful propagation of plants that do not reproduce via seeds, such as bananas, thereby highlighting its significant role in modern agricultural practices. Combining various clones into a single tree can lead to innovative varieties, such as fruit salad trees. This practice showcases the creative potential inherent in grafting techniques. Tissue culture is an effective approach for rapidly producing thousands of identical plants. This method greatly benefits commercial agriculture by guaranteeing high yields and consistent quality. While clonal propagation aids in achieving desired traits, it can inadvertently lead to monoculture, which diminishes genetic diversity. This scenario poses risks, notably increased susceptibility to diseases affecting entire crops. The propagation process can also contribute to disease transmission, making it essential to sustain healthy plant stocks. Higher costs often complicate the establishment of more digital cultural labs. Plant cultivars originate from the careful selection of seedlings, which plays a pivotal role in maintaining genetic diversity. Understanding the foundation of these cultivars is crucial for effective propagation. Mutations, whether spontaneous or induced, significantly influence genetic changes in plants. These alterations can yield new characteristics that enhance the cultivation process. Chimeras represent a fascinating type of genetic variation featuring mixed tissues with distinct genotypes. This phenomenon can result in plants exhibiting multiple colors or traits, contributing to their uniqueness. Transposons, genetic elements capable of shifting positions within chromosomes, contribute to variations. They can induce unstable traits, such as color changes in flowers or leaves, highlighting the dynamic nature of plant genetics. Somoclonal variation arises from tissue culture techniques, which can lead to the emergence of new plant varieties. This approach involves cloning cells that may showcase unique traits. Stable plants yield consistent copies through vegetative propagation, whereas unstable plants may exhibit varying colors, such as pink from a white variegated parent. This distinction is pivotal for growers. Transposons in spider plants exemplify how genetic variations manifest visually, resulting in traits like streaks or dots. This observation underscores the significance of genetic stability or instability among plant species. Chimeras emerge from specific cell layers, leading to mutations in plant tissue that can result in variations such as paraclinal and mericlinal types. Comprehending these variations enhances plant breeding practices. There are various types of chimeras, including sectorial, where mutations are widespread throughout the plant, and paraclinal, where only part is affected. Each type can significantly influence propagation outcomes. Variegation in plants often results from chimeras, leading to striking mixed color patterns due to either the presence or absence of chlorophyll. This characteristic can enhance the visual appeal of plant varieties. Viruses may also induce variegation in plants, which is distinct from the genetic variations associated with chimeras. The mosaic virus, for instance, produces a yellow and green leaf pattern. The significance of maintaining new cultivars cannot be overstated. Proper care and attention ensure the stability and retention of the desired traits in propagated plants over time. An emphasis on chimeras reveals their critical importance in plant propagation. Gaining insights into chimera formation can lead to enhanced practices in the development of diverse plant cultivars. Lastly, various methods of plant propagation are introduced, underscoring the importance of grasping both sexual and asexual propagation techniques for successful plant development.